And now I'd like to introduce our final speaker for the session, Pankil Gawade. He's a PhD student at Helmholtz Munich, and his work uses artificial intelligence to tackle antibiotic resistance, one of the biggest threats to global health. And today he'll present AMPFormer, a foundation model for discovering new antimicrobial peptides.
Thank you. Thank you. So today I'll be talking about a peptide foundation model for antimicrobial discovery. Antibiotic-resistant infections are supposed to kill around 10 million people annually by the year 2050 if no new measures are being taken. Antimicrobial peptides are a solution to this problem because they are short chains of amino acids, usually less than 50. So they are also less toxic to mammalian cells, and it's harder for bacteria to develop resistance against them, mainly due to their mechanism of action. And that's why they're also a good alternatives to conventional antibiotics. Why it is difficult to make these peptides is because of the astronomically large peptide space. And it is estimated to be around 10 to the power 65. I mean, the number doesn't make sense to me even, yeah. So traditionally, people have been attracted towards AI methods. So typical AI-driven AMP discovery pipeline consists of, let's say you take a peptide database, and then you train a generative model, which is expected to model an AMP distribution, of which then you can sample novel AMPs. Or you can take a large library of biological sequences, which can be proteins. You can bin them to get putative AMPs, of which you can pass them through a predictive model, so which can discriminate between an AMP or non-AMP. This offers limited controllability. It's sample inefficient, and it's unreliable when the data is scarce, which often leads to expensive wet lab validation and low experimental hit rate. So we present AMPFormer. In AMPFormer, we combine the two settings, that is prediction and generation, in the same model. So we take the initial pre-training data set of about 3.7 million peptide sequences, and then we do this joint pre-training where we use alternating attention and task-specific heads. So there are two heads responsible for generation, that is next token generation and reconstruction, and rest of the 39 heads for property prediction. So it's charge, hydrophobicity, aromaticity, TPS, etc. So this model does not only does generation, but property prediction using the same backbone and shared weights. So once you have this model, you can fine-tune it on actually meaningful, experimentally validated sequences, data that you have. And this data usually consists of different bacterial strains and their measured MIC values. So you can fine-tune AMPFormer to get extended performance from it. So this model not only is aware of sequences, but also descriptor information. And this is a novel architecture giving you synergistic benefits of joint generation and property prediction. And this has already been proven for molecules in this paper. Additionally, we introduce self-improving active learning loop with joint test time reasoning, where from the left, you start with AMPFormer, which is fine-tuned. So it generates sequences, and it also predicts the properties for given bacteria. So we calculate a score based on these predicted properties, and then we use this joint test time reasoning, where for example, you start with a root node at the top, and then you run stochastic beam search, and you sample multiple sequences of peptides. So each leaf node in this case is a peptide, and then you score these peptides using the scores, Rx. You choose the best possible peptide, and you trace back till the blue point, and then you run stochastic beam search again. You do this till you reach the maximum sequence length. We do this, and then we get sequences of peptides, which are novel and unique. Once we get these, we pass them through an in silico oracle, which is an ensemble of multiple ML models. And then we get predictions from these from the ensemble, that is oracle, and then we again calculate the score, Rx, and then we use a top K filter to filter out best possible peptides based on the score, let's say top 10. We append these peptides to fine-tuning data set, and then we fine-tune the model again. We do this a couple of times till we get best possible peptides, and then we send them to experimental validation. So this is not only sample efficient, but it also offers you controllability even when the data is scarce. For experiments, we begin with broad spectrum activity. So the aim here is to kill all the broad spectrum bacteria. For second experiment, we do gram-negative selectivity, where the aim is to kill gram-negative and not kill gram-positive. The third experiment would be to kill gram-positive bacterial strain and not the gram-negative ones. And the fourth is to spare commensal bacteria, but not, spare commensal bacteria, but kill the broad spectrum, because commensal are the good bacteria. So before beginning with these experiments, we do a sanity check of the generated peptides. Do they even make sense? So we benchmark AMPFormer against multiple models, and we see that we outperform the other models in biological fitness and all the predicted activities from different external classifiers which were trained on different data sets. This was again shown in the paper below. So once we have the sanity check done, we then move to the experiment 1, where the aim is to kill all the broad spectrum bacteria, and this was done in vitro as well. And here, all the 25 peptides that we tested in the wet lab killed multidrug-resistant bacteria, and especially one peptide was active against all 17 considered bacterial strains. So in the plot below, you can see on the x-axis, it's MIC threshold, MIC being the minimum inhibitory concentration, that is minimum concentration needed to kill a bacteria. And on the y-axis, you have AMP success rate. So in this plot, to be on the top right would be the place, and AMPFormer achieves it with as low as 16 MIC threshold, by far overcoming all the other models. So once we are done with this experiment, we move to the experiment 2, 3, and 4, which is gram-negative, positive, and commensal selectivity. And this was done in silico at this moment. So here, the blue is the gram-negative, which we want to kill, and the red would be the gram-positive, which we want to save. So in this case, PMIC, the higher, the more potent. And we see this iteration 1, 2, 3, 4 indicating the active learning loop that we discussed earlier. From iteration 1 to 4, so iteration 1, you get initial signal, and then eventually by iteration 4, you see the maximum possible separation, giving you peptides which are possibly likely to kill gram-negative and spare gram-positive bacteria. Then we move to experiment 3, which was killing gram-positive and not gram-negative. This is difficult to begin with because of low signal in iteration 1, but also because biologically killing gram-positive bacteria inherently allows you to kill gram-negative as well. So you start with iteration 1 with very less signal in the active learning loop, and then by the iteration 4, we are able to get meaningful difference between these two means, indicating that we get the peptides which could possibly kill gram-positive and not gram-negative. And the same goes for experiment 4, where we aim here is to kill broad spectrum and not commensals. So iteration 1, we start with difference, but eventually, as we move into the iteration, the difference increases. So we select best possible candidates from each of these cases, from each of the iteration, not just the final iteration, and then we have sent it for experimental validation. So in conclusion, we provide you with a peptide foundation model, which can be used by community for property prediction, generation, or optimization of sequences. We also introduce self-improving active learning loop with test-time reasoning, which can be used for targeted bacterial species-specific control generation and property prediction. And what's remaining at this point, which is in process, is the in vitro validation for gram-negative positive selectivity, as well as the in vitro validation for commensal activity. So stay tuned for the exciting results, and thank you very much.
Thank you. Are there questions? I see one in the very back there. Please raise your hand again. Yeah.
Hi, nice talk.
Thank you.
I was wondering, the peptides that you generated using your model, how novel they are in terms of sequence similarity or the scaffolding, like the question in the direction that are you catching or generating the sequence that you already showing the model during training, or they're truly novel ones? Thank you.
So to answer this question, I would just mention you some email exchange we had with our biological collaborator. We sent him peptides initially, the ones that were experimentally validated, and he replies to us that these are peptides that are not possible to be conventional antibiotics. Like, they don't look like conventional AMPs, but they turned out to be active against all the bacteria that we tested, and all 25 of them. So I would say they are quite novel in general.
Okay. One further question in the front here.
Yes, following up on this, what about the toxicity of these novel peptides? Is it something that you think the foundation model already took care of? And if it has information to the toxicity of peptides, or is it something that you will test experimentally, already tested before?
So we have to test it experimentally. But what we can do is we can also, once we have this pre-trained model, which is very informative about sequences and descriptors, we can fine-tune it on experimentally validated toxicity dataset to make a classifier or predictor, and then filter out the sequences which will not likely be toxic.
Thank you.
Yeah.
Okay, great. If there's no further questions, then thanks again for your presentation.